Okay, good afternoon everybody. Please, I want your attention to start the symposium of this afternoon. Okay. As I said, let's start this afternoon this symposium focused on aging in healthy and in pathological brain. Then, as the title said, we will see how the crossroad between aging and neurodegeneration can be monitored and used to diagnosis of neurodegenerative disease, and perhaps also to develop new therapies or develop the individual medicine. The first talk will be given by the invited speaker, Nicolai and Franzmeier, with a lecture titled Using Multimodal Neuroimaging to Understand Alzheimer's Disease Progression. Dr. Franzmeier is a junior research group leader in the Ludwig and Maximilian Institute for Stroke and Dementia Research at the University Hospital in Munich. His research focuses on understanding the brain mechanisms related to the progression of Alzheimer's disease pathology and on those mechanisms that might be important for the development of dementia despite the presence and progression of AD pathology. Dr. Franzmeier, the floor is yours.
Thank you. Yeah, thank you for the introduction. I hope everyone can hear me well. Thanks to the organizers for inviting me, especially at David, of course. And I'd be very happy to give an introduction today on how we in our group in Munich, and of course the worldwide community, try to use multimodal neuroimaging and biomarkers to understand the progression of Alzheimer's disease. So, since not everyone here in the room is obviously a researcher in Alzheimer's disease, I would like to start with a brief definition of how we define the disease today. So we understand it biologically, defined by a cascade of pathological brain changes. You see here a plot time versus abnormality of a biomarker. And we know that Alzheimer's disease potentially starts with the brain-wide deposition accumulation of amyloid pathology. That can be roughly 15 to 20 years before people develop first symptoms, and that is then followed by the gradual spreading of tau pathology from somewhere in the temporal lobe, typically throughout the brain, and we'll hear more about that later on. And together, these two processes, if they've happened for long enough, lead to neurodegeneration, as we've heard this morning from our plenary speaker, giving rise to a spectrum of cognitive decline from cognitively normal to mild cognitive impairment, or MCI, up to the level of full-blown dementia. And I'll try to cover two different major topics today. First of all, understanding what drives and what modulates the progression of the primary AD pathology itself. And second of all, what drives the clinical manifestation, especially clinical heterogeneity. And here with a particular focus on cognitive reserve and resilience. And to address these different topics, we, as the title of my talk says, we use PET imaging mostly and fluid biomarkers to a certain degree, which we combine heavily with connectomics and to a certain degree genetics. And I would like to start from the end of the disease, which is the clinical phenotype. And here, it has been known for 20 to 30 years roughly that there is a large variability in the clinical manifestation of the disease, meaning that some individuals can better cope with brain pathology. They maintain cognition despite having the disease. We've heard that this morning in Michael Valenzuela's talk already. And this has been for ages associated with factors like lifetime experiences, as we heard this morning, education, intelligence, physical activity, all of which have been shown to attenuate the effect of brain pathology on cognitive performance. And this is not exclusive for AD. You find that in every neurological condition that you potentially could think of. But what is unknown, or what has been unknown so far, is how does the brain actually do that? So how does the brain cope with pathology? And in order to study this, we've focused on functional MRI, functional connectivity, within a particular brain network, which you can see here, which is the cognitive control network. It's kind of a domain general network that is important for a variety of cognitive functions. And we got particularly interested in that network because previous studies have shown that if that network is more globally interacting with the rest of the brain, that correlates with these typical reserve or resilience proxies, such as education or intelligence. So we were simply asking the question, does the control network support reserve? This is 5 years ago, roughly, that we conducted the study where we combined fMRI to determine how globally connected the control network is with FDG PET in a sample of patients with a sporadic Alzheimer's disease. So all of them had amyloid pathology and were mild cognitively impaired. And these people typically develop synaptic dysfunction, glucose hypermetabolism in the precuneus and posterior cingulate, which typically leads to or is associated with memory deficits. And you can see here in the orange line that in fact those people whose control network is not as densely interconnected with the rest of the brain, they show a memory deficit as metabolism goes down, whereas those people with high connectivity don't really seem to care. So this was the first indication that there is a buffering effect implemented by the control network. And then we tried to go one step further to go in a very clearly controlled sample with autosomal dominant AD. So all these people carry a mutation which drives the development of the disease. And in those people we can track the changes in the biomarkers from 20 years before up to 10 years after symptom onset, which is based on the age of onset of the parent that they have inherited the mutation from. And you can see that biomarkers become abnormal as we expected, but if we project in cognition, depending on having low or high control network connectivity, you can see that the dashed line gives you a shift of the clinical trajectories towards a later time point. Overall suggesting that the control network has something to do with reserve, but we don't know the mechanism by which it does that. So the cognitive neuroscience literature has shown for several years now that the control network is kind of the organizer of brain activity in a very simplified term, and that it drives efficient network organization, especially during task demands. So what we can quantify as brain network segregation, so how well is the brain organization organized into different modules. And so we've looked at whether segregation is the actual effector of reserve and resilience effects. And again, we used two independent cohorts of autosomal dominant and sporadic AD. And here you see that people with high brain network segregation show less cognitive deficits than are expected compared to individuals with low network segregation. Very similar, so in sporadic AD, the y-axis is inverted here. At a given level of tau pathology, those people with higher network segregation show less cognitive deficits. So overall, we believe that this classical model of reserve and resilience that puts protective factors at center stage can be extended putatively by a mechanism, which could be the control network and brain network efficiency, and we've really published many papers on that, and others have started replicating our findings as well. And with that, I would like to close the chapter on reserve and resilience and move forward to what drives the development of the disease in the first place. So what we've seen now is kind of the end of the spectrum, so the clinical phenotype, but what modulates the development of the pathophysiology itself. And as I said initially, Alzheimer's starts with, or that's what the model assumes by now, starts with amyloid deposition and amyloid accumulation. And even Alois Alzheimer in his 1906 publication has already shown that he found activated microglia in the brains of the patients. And we now know that this is happening relatively early on. And microglia are the brain's resident immune cells which react to any pathological insult. So they're cleaning cell debris, for example. And it has been shown already, as you can see here on the right, that they also encapsulate the initiating amyloid plaques and kind of build a physical barrier around them. So The question is that a protective or a bad effect overall, which might lead us to a new therapeutic approach? And how can we measure that activation in living patients? So part of that microglial response is driven by a molecule called TREM2, which is a surface molecule which is shed, and we can measure the soluble part of it in the cerebrospinal fluid, or CSF of patients, which was done by a former colleague of mine who's now actually working here at Barcelona Beta, Marc Suarez. And he looked at an autosomal dominant and sporadic AD cohort and mapped the trajectory of soluble TREM2 in the CSF. And he could show that there is basically an initial increase in the microglial response that we observe, which is then vanishing again later on. Very similar so in sporadic AD patients. But the question is that something good or bad happening? So is inflammation helping protect the brain, or is it actually accelerating the development of the disease? So we started looking at this in mice, because this is a very controlled setting where we combine TSPO PET imaging, which is a microglia marker, so it binds to TSPO, which is expressed in activated microglia and macrophages, and plotted that against the longitudinal rate of amyloid PET accumulation. And this is just naturally occurring variability in microglial activation in mice that develop amyloid pathology. So they have lots of mutations that are knocked in. And you can immediately see that those mice with higher microglial activation at baseline show slower accumulation of amyloid over time. So this was preclinical evidence, which we then took to clinical data in AD patients where we had amyloid PET longitudinally captured over time in combination with soluble TREM2 levels from CSF. And if you typically plot baseline amyloid PET uptake against the longitudinal accumulation rate, you typically get this inverse U-shaped association, meaning that there is an initial dynamic increase in amyloid accumulation, which is then later on saturated. So this is typical and has been reported before. But however, if you split up that curve by having high or low TREM2 levels at baseline, you see that the orange line reflecting those guys with higher microglial activation is actually flattened, so there's less amyloid accumulation over time. So we see slower amyloid accumulation in those people associated with increased microglial activation. And what does that mean for the clinical trajectories of those patients? So we've looked at this again in a cohort with longitudinal data up to 10 years of cognitive follow-up, and all of these patients were clinically impaired, so at least MCI or AD dementia, all amyloid positive. And ADAS-13 is a global cognitive scale. The higher it is, the more abnormal the score is. And you can again see that there is an interaction with TREM2, such that people with higher TREM2 decline less fast over time. This was longitudinal data. And then, but this was a clinical population. So what is if we move into an at-risk population? Here, carriers of the ApoE4 gene, the most striking risk factor for developing a sporadic AD. And here you can see these guys are basically split up again by their TREM2 levels, plotted against the longitudinal rate of cognitive change. The more positive it is, the greater the worsening in those patients. And again, people with higher TREM2 levels, higher microglial activation, show less of a decline in cognition over time, overall suggesting that, at least to a certain degree, microglia might be protective, but this is certainly an oversimplification, and we need to understand the full complexity of which microglia play into the overall disease trajectory. And with this, we would move now to the next, and what I believe is the most striking pathological brain change in Alzheimer's disease, which is tau. Tau is much more closely related to the onset of symptoms, as we'll see in a minute. And tau is, in its physiological form, a highly abundant protein in neurons, called microtubule-associated protein, encoded by the MAPT gene, and helps stabilize those little highways within the axonal compartment of the cell. However, if it becomes hyperphosphorylated in Alzheimer's disease, these little p-tau, for phosphorylated tau, seeds start forming, they start clumping together, forming prefibrils and tangles, and basically the whole neuron falls apart afterwards. And what is very important for the rest of the talk, these first initial hyperphosphorylated tau seeds have been shown to travel across synaptic connections. So there's a supposedly prion-like mechanism which is mediated by the brain's wiring circuitry, so to say. And why is understanding the spreading so important? Because tau is really the key driver, at least what we know now, for neurodegeneration and ultimately cognitive decline. So we previously, Heiko Braak and his wife, they've established this post-mortem tau staging, which we can now use or apply to tau PET imaging. So where we have a radioligand that binds to tau fibrils in the brain. And when we quantify the Braak stage in AD patients, then we can basically quantify how severe is the degree of tau pathology in a given individual and plot this against the rate of cognitive decline here for the mini-mental examination scales or global cognitive measure. And you can clearly see the more advanced people are on tau, the faster they decline cognitively. And that effect is much stronger than you would see it for amyloid. So the idea that we now have is to understand the progression of tau pathology by bringing together connectomics to understand the brain's wiring diagram and tau PET for bringing those together to understand whether tau really spreads across interconnected brain regions. So that's the basic idea of that whole endeavor. And in the first two studies where we did this, we combined, as I said initially, longitudinal tau PET imaging and resting state fMRI-based connectivity in 2 independent samples of Alzheimer's disease patients. And when I mean Alzheimer's disease, I always say that they're amyloid positive on a biomarker level. And using this 400 region of interest brain atlas, we first of all quantified how well are the brain regions interconnected. And in a second step, we took their tau PET levels and quantified how similar is tau accumulation between any 2 given regions, which is simply the covariance in tau accumulation. And if you bring these 2 together, you can see that brain regions which are functionally connected to each other show a more similar rate of tau accumulation over time, telling us that connectivity is somewhat informative about what is happening in terms of tau accumulation. And we were able to replicate this then in the Swedish BioFinder cohort as well. So based on this very simple association, we were wondering whether we can use the brain's connectomic roadmap to actually predict the pattern in which tau travels throughout the brain. So we took again 2 independent samples. You see that's a reoccurring scheme. We are always trying to replicate findings to be certain that we're not reporting false positives. And we used again 2 samples that span the entire preclinical to clinical AD spectrum, so from cognitively normal, mild cognitive impairment, to dementia. And you see over the treatment course that tau, which is more red, becomes more abnormal, as we would expect it, of course. And then we can take this cross-sectional variability to build a tau spreading sequence. So we define which brain regions become abnormal consistently very early on, and which brain regions show late tau accumulation. And even more importantly, we can define those brain regions which are potentially the ones that become abnormal first, which are typically somewhere in the inferior temporal lobe, most of the times the entorhinal cortex. And then we used fMRI again to map out where are these regions here in green connected to, which you see here on the surface plot. And just by visual comparison, you can see that the spreading sequence, where more yellowish regions become abnormal earlier, highly align with the connectivity of those brain regions. And in fact, if you correlate those with each other, you see that the epicenters and how they are connected to the rest of the brain give us a very good explanation in terms of which brain region becomes abnormal when, again, highly consistent across 2 cohorts. But that was cross-sectional data. What is, if we try to model that longitudinally using individual patient data? That means we take a given patient's tau PET when he comes into the clinic and define that region which shows highest tau pathology at that point in time. That's our epicenter. And then we map the connectivity of that epicenter to all the remaining brain regions and put them like pearls on a string and cut them in 4 different groups depending on how closely they are connected to the epicenter. In essence, we're trying to define here 4 regions that are classified based on how well they're connected to the site of tau onset. And we would expect that these regions here for Q1 or quartile 1 show the fastest tau accumulation over time and that this effect vanishes. And you can see this gradient clearly here in 3 independent samples where this Q1 region is always the one accumulating tau fastest over time. And with this way, we can really zoom into a patient's brain and predict where is the pathological change occurring. And this is important because we have now a sensitive measure to define a potential endpoint in a clinical trial on a biomarker level, because if you compare the metrics that we're suggesting against all other established Braak staging or whole brain region of interest readouts, we're more sensitive. And measuring a change more sensitive means that if you define that as an endpoint, you need less participants in order to determine a treatment effect, and we're currently trying to implement that exploratorily in clinical trials together with Eli Lilly. So this was giving us a global picture on how tau and how tau brain networks and tau interact with each other. But we know that not every brain region shows the same connectivity pattern, but that what I mentioned initially already, that the brain is organized into modules and that we have hubs which are densely connected and we have regions which are non-hubs which are poorly connected. And here we were wondering whether if tau hits a hub region, do we actually observe in a COVID analogy something like a super spreading event, that the spreading is really accelerated and fast, whereas if it is happening in a non-hub region, then we anticipate tau spreading to be relatively slow. So we tried to test this by combining again fMRI with tau PET, and we took fMRI data from 1,000 healthy individuals of the Connectome project, and first of all defined which brain regions are these globally connected ones. So we just simply mapped out how strongly connected is one particular brain region to the entire rest of the brain. And then built a gradient that scaled linearly between hubs and non-hubs. And we then mapped the individual tau PET patterns to this hub map. I won't go into the mathematical details here, but basically that gave us a single index telling us whether tau was more biased towards hub regions, as in this individual, where you have more tau here in the superior parietal lobe, whereas if tau is more biased towards non-hubs, typically here in this inferior temporal lobe. And what we could clearly see here again in ADNI, that the more tau was biased towards hub regions at baseline, the faster those people were accumulating tau subsequently, suggesting that hubs are really accelerating factors for the biological development of the disease. And in Biofinder, we found again a very similar result pattern, here only in symptomatic AD patients, because those without symptoms simply had no tau pathology at that point in time. And I'm sorry that the axes are missing here. That's a Windows Mac incompatibility, I guess. And even further, we found that people at a younger age who were symptomatic showed more tau in their hubs. And again, in both samples, and that is a very nice reflection of a previous study conducted by a colleague in Gothenburg, Michael Schöll, who showed that people with early onset AD, so typically before the age of 65, these guys develop symptoms, show a more neocortical superior tau PET pattern, exactly in those brain regions which are hubs in our sense. And this could mean two things. That first, these hubs are, of course, as you've seen in the initial part on reserve, highly important for cognition. So if they're functionally impaired, probably there's more of a clinical loss as well, but in a pathophysiological sense, we also believe that these hubs themselves accelerate the development of the disease. So we put this together in a mediation model and could actually show that younger age relates to faster tau accumulation, and that this effect is to a large degree mediated by having tau in these subregions that together leads to faster cognitive decline over time. But can we make more out of this brain modular network structure? So is there more of an effect beyond just telling us where tau is starting and where it is going? Because I've meant very initially that there are high inter-individual differences in the way that brain networks are segregated from each other, here highlighted by these three different modules. And we've shown previously that individuals with higher brain network segregation actually show less cognitive deficits as you would expect them to have. But is that a pure resilience effect, or does a higher segregation of brain networks actually also attenuate the interregional spreading of tau pathology? Because there are simply less ways or less shortcuts between modules that tau could go. So a PhD student in my group, Anna, has recently looked at that, and that paper is coming out hopefully next week or so, where we've again looked at longitudinal tau PET across a group of AD spectrum patients, which was combined with resting state fMRI data. And here's just an illustration of how their brain network segregation here, color-coded, looked for people with below median segregation and above median segregation. You can clearly see in the above median group that there is a better color separation, suggesting that the modules are within and among themselves better organized. And then we looked at the entorhinal cortex, this is just one of the analyses in the paper, as the putative starting site or epicenter of tau pathology, and its predictive value for how is tau accumulating in the rest of the brain subsequently. So simply you plot tau PET in that region against the accumulation rate in the remaining brain. And you find that anticipated positive association such that if people have higher tau at baseline, there's faster accumulation in the rest of the brain. But there's also an interaction with brain network segregation such that individuals who have more segregated brain networks do not accumulate that fast, which is an indirect evidence that more segregated networks may act as a functional barrier, keeping tau confined to where it actually is located at the moment. But this was a lot of data on brain networks and tau spreading, but one question that I haven't touched yet is what initiates tau spreading in the first place. And we've seen that cascade initially, that amyloid comes first, which is followed by tau, but every one of us literally at a high age develops fibrillar tau pathology in the entorhinal cortex to a certain degree. But in the absence of amyloid, it typically stays there, so it doesn't go out. But only when we develop neocortical amyloid plaques, then we see tau traveling out of the medial temporal lobe. And this is, I must say, specific here for Alzheimer's disease tau. There are other tauopathies, primary tauopathies, but there is a different molecular composition of tau. So here we're talking about 3-4 repeat tau, if you want to have the precise nomenclature. And amyloid really triggers the spreading of tau. So how is that happening? That's one of the long-lasting questions. And we were looking then back at more recent biomarker data that combined fluid biomarkers of phosphorylated tau with fibrillar markers of tau, so PET. And if you plot both p-tau, so that's measured in CSF, and fibrillar tau via tau PET against each other, or against amyloid PET, so here fluid metamo PET SUVR, then you see that CSF or fluid markers of tau become abnormal before tau fibrils build, which makes sense from a pathophysiological point of view that the seeds, which we can measure in CSF, potentially precede the formation of tangles. And the pathophysiological model that we have in mind here is that amyloid accumulation triggers the initial phosphorylation and formation of first tangles, and that this leads to a secretion of p-tau at the synapse, potentially driven by neuronal activity because amyloid drives neuronal hyperactivity. And that this release of p-tau at the synapse then leads to the subsequent spreading to the next neuron, giving rise to neurodegeneration and cognitive decline. And importantly, we can measure approximately the secretion of p-tau at the synapse by using CSF p-tau data. So we simply asked here, is p-tau actually the culprit that drives amyloid-related tau spreading? So in the biofinder cohort here, these are 170 roughly individuals, all amyloid positive. We looked at longitudinal tau PET and whether amyloid or p-tau were predicting the accumulation rate in tau. You can see amyloid does predict it, yes, but it's outrun by p-tau, so there's a much stronger effect. And in fact, if you put both into the same model, then you see that the effect of amyloid completely vanishes, and only p-tau explains what happens subsequently in terms of tau accumulation. And you can formally then put those together in a mediation model, and here we found for those brain regions highlighted, which are the typical ones accumulating tau, that the effect of amyloid on tau accumulation is more or less to a large degree mediated by increases in soluble tau, suggesting that this is really the culprit driving the spreading. And we tried to embed this then into our connectivity-based spreading model. So we again defined for each individual the epicenter of tau pathology, mapped the connectivity of that epicenter, and plotted against the rate of tau accumulation over time, as you can see here. So the closer regions are connected to the epicenter, the faster they accumulate tau, which is reflected in that negative slope of that association. So that slope index, which is a beta value, is an index of how well tau accumulation aligns with the brain's connectome architecture. And you can see overall that this is negative for the group of AD patients here. And if you look at this in conjunction with p-tau, then you actually see the more abnormal p-tau is, the faster these guys show connectivity-mediated tau spread. So these are the beta values that reflect this slope here. And this was relatively complicated, so we can illustrate that more simply by bringing together that initial analysis that I've shown you previously by looking at these 4 different regions of interest that we just cut out of the brain, where we anticipate this brain region with closest connectivity to the epicenter to show fastest tau accumulation. And we see again that gradient. These are 2 different samples from the ones you've seen before, and if you break them up by having normal or abnormal p-tau or high or low p-tau levels, you see that only those guys with high p-tau show that accumulation, suggesting again that p-tau is a necessary precondition for tau actually to spread from where it is located to the next region, which suggests that p-tau is a potentially very interesting treatment target, and we're trying our best at the moment to understand how amyloid drives p-tau increases. And again, microglia may play a role, and neuronal activity is a hot candidate here. And lastly, I would conclude with some of the work that we did on understanding what further modulates tau spreading. Here looking at genetic factors, most importantly BIN1, which is a highly abundant risk factor in a gene encoding for a protein that helps with a vesicle curvature. And roughly half of the European population carry that risk gene. And after ApoE, considered, depending on the source you look at, the second most important risk factor for sporadic AD. And preclinical data has related that BIN1 SNP to tau pathology, because it was supposed to help with the endocytosis of tau-containing vesicles. And so we, for the first time a couple of years ago, looked at that SNP, that's what it's called, 744373, between risk and normal allele carriers, and simply looked at their tau levels across the entire brain, so again picking up this Braak staging scheme. And in yellow, you can see that basically across the entire brain, there's higher tau levels in those guys carrying the risk allele. Whereas if you look at amyloid, there is no such effect. They're even a bit slightly, if you look at the median, lower. So here we have a genetic risk factor that is specifically associated with tau. And if we look at this further in the context of amyloid and combining it with longitudinal tau PET data, and then we could see that people at a given level of amyloid show faster tau accumulation over time. But if you break that plot up by having that risk allele in yellow versus the normal allele in blue, whatever that color is, to be honest, then you see that those guys with the risk allele show faster tau accumulation over time. And therefore, we are currently trying to team up with preclinical researchers to better understand what is the molecular consequence of carrying that risk allele to tell us more about the mechanism here. So with that, I'm already at the end. So just a summary of what we are trying to look at in our group in Munich, together with our collaborators. We're really trying to use imaging biomarkers to understand mechanisms of the disease as good as we can with imaging data, which is always just indirect evidence, to predict the progression of the disease while taking into account factors that potentially modulate the disease. And with that, I'm at the end. I would like to thank, of course, my team in Munich, many collaborators, in particular Oscar Hansen, Rick, Austin Koppel in Amsterdam, and many, many others who are not here on the list. And thank you for being so silent and paying attention. And I hope that I didn't confuse you too much, and I'm happy to take questions if there are any.
Thanks very much, Dr. Franzmeier. Just a remember to the people that are on the stream that if you have questions, you have to write to the chat, and I will read for the speaker and for the audience. Then we have 5 minutes for questions.
Hi, thank you very much. It was Nico, great talk, fantastic.
Thank you.
Really enjoyed it. So at some point you seem to imply that you said that because of the findings that you have had, that brain activity and brain connectivity is like a driver of, maybe related, I don't know exactly the direction clearly, as you show with the pathophysiological changes in tau in particular. So do you think that therapies that could directly tap on changes in brain connectivity and activity, I'm thinking about pharmacological therapies, but also maybe non-invasive brain stimulation, can be, I'm not saying like therapy to cure maybe, but at least to modify a bit somehow, potentially.
I see where this is heading. And I think we need to establish a couple of more links before we can make a proper conclusion here, which is link between neuronal activity and p-tau. I think that is important. We talked about this yesterday in the evening. But if that turns out to be true, then this brings in neuronal activity at center stage for modulation, which we can do using repurposing old anti-epileptics. And we're planning actually a clinical trial, an exploratory one, looking at levetiracetam and seeing whether this does anything to amyloid-induced hyperactivity and has any effect on p-tau. That's highly exploratory, of course, but this would basically tell us neuronal activity is important. In terms of modulating brain activity using brain stimulation, here you are clearly the expert. If you can define a target, so a state in the brain that you want to foster and induce, such as brain network segregation, maybe. I have no idea whether or how this would work, but if the associations that we are showing here are, even though most of it is cross-sectional data, are kind of causatively linked, then modulating one factor should have an effect on the other, but that's the gold standard of any experimental study. If you modulate something, look at the outcome, but I would, from a gut feeling, think, yeah, that might work. Michael?
Great talk. My question relates to, there's a very nice paper by Pallàs which showed very longitudinal, I'm talking about 7-8 years of atrophy changes from the time of diagnosis, and then segregates people into, I think it was 4 or 5 clusters. And one of the most common clusters is where the neurodegeneration only stays in the limbic area, it doesn't go cortical, where other patterns, it's limbic and then cortical, a la Braque. Mm-hmm. So I'm wondering if you've seen any of that type of longitudinal cluster phenotypes in the tau.
Yeah, data. Definitely. I didn't show this, but it's part of the Science Advances paper. There is large heterogeneity, so that Braque staging that we're commonly applying accounts for 70%. The rest is not following that trajectory. There's asymmetry. We know that there are clinical variants which do not follow that pattern, like posterior cortical atrophy, primary progressive aphasia. But these are extreme phenotypes, but within the regimen or the range of normal, what you would call normal amnestic Alzheimer's disease, there's a large heterogeneity in tau, and tau develops closely with, or matches closely with the development of brain atrophy. So we see brain regions, that was a paper by Renaud Lajoie from UCSF a couple of years ago, where you have tau at a given point in time predicts where you will degenerate in terms of gray matter volume next. And I would closely, or hypothesize that the clusters that they have been seeing are to a large degree driven by tau and potentially also neurodegenerative co-pathologies, which we can't measure by now. Yeah, but this is clearly something we see in tau.
Some more questions? Here.
Nice talk. I was wondering, because here you showed nicely this link between beta amyloid plaques and tau spreading. So what's your opinion about all of these anti-amyloid therapies in humans not showing really good results? Do you think it's because it's maybe done too late? What's your opinion about that?
Yeah, I have this analogy of if you think of an avalanche that is caused by a person which is walking along with skis on top of the mountain, and if the avalanche has reached and destroyed the village, then telling the skier up there to go down probably won't save the village because it is destroyed already. So it's a matter of timing. I think amyloid, I do not question the amyloid cascade at all, but we have to think about when are the consequences happening, and if you want to target amyloid and prevent anything that comes afterwards, then you need to move in extremely early, and no one knows when. And the trial results from the DIAN trials unit with gantenerumab, and I don't know the second drug that they've used, they didn't look too good, did they? So potentially they have moved into late as well. Now lecanemab looks better, donanemab looks better, aducanumab looked at least in one study good and the other one bad. I would say ultimately we will need anti-amyloid trials, but we will need combination therapies. And next year probably the clinical reality will be anti-amyloid therapies all over the place. And we need to add on top of that with either targeting tau or what Michael showed in the morning, targeting neurodegeneration, which is most closely linked to cognition. Yeah, so that's my general opinion. I'm a bit skeptical also about the clinical effects of these anti-amyloid drugs. Half a point in CDR, is that enough for the patient to realize anything? From a clinical perspective, I would be skeptical, at least, especially given the pricing of these drugs and the side effects, which are can be quite strong.
There is another question there.
Excellent talk, so I enjoyed very much. So my question is, any correlation between the soluble TREM2 and tau spreading?
Yeah, we have that under revision. So yes, there has been recent work that showed that TREM2 helps keep amyloid in its barrier, but at the same time can help increase p-tau and tau phosphorylation. I'm not sure about the molecular pathways, but that has been a paper by the David Holtzman group, which are really the spearheading the TREM2 research field. And we have found a very similar thing using biomarker data. So when we go in very early, when people are CSF amyloid, but not yet PET positive, so there's a similar distinction between fluid and fibrils, then TREM2 is linked to an increase in p-tau. So there it's very early on, we believe at least, not a good thing to happen, whereas later on, when people have symptoms and severe pathology, so when there is enough stuff to clean up, more or less, then microglia become protective. So I think the therapeutic window for microglial activation, and we've just discussed this before, needs to be clearly defined. So it's definitely not a good idea, at least that's my feeling, in a person which has amyloid but no inflammation yet, just to kick off the inflammatory cascade. That will do something to you, I guess. But it's very complex, and we're just starting to understand it.
Thank you.
Okay, thank you very much, Nicolai. We have to move to the next talk.
Yeah, thank you all.
The next talk will be given by Lluís Fuentemilla. Dr. Fuentemilla is full professor in the Faculty of Psychology in this university and is member of the UBneuro. Is PI of the group of brain plasticity and connectivity, language, memory and reward group. And the focus on his research aims to understanding how the human brain supports the formation, the consolidation and the retrieval of everyday life experiences. The floor is yours.
Thank you, Mercè. Thank you, organizers, for inviting me. Thank you for setting up this platform, to an ambitious platform, right? To put together many aspects of the brain and how this impacts in disease. And just to start with this, I just want to briefly mention that for for your expectancy, I'm not going to talk about Alzheimer's disease, just to make sure that I'm going to be the point. The other sad aspect of my talk is that I'm going to ground my findings on something that was known at least 150 years ago. So putting all these things together, my ambition is at the very end of the talk, I could kind of like grill some sort of like motivation or some sort of insights into what things can be done in the years to come, right? So just to make these 2 points clear. So what we know from psychology is that somehow memories, the fate of the memories is to disappear over time. And that was shown by Ebbinghaus. It's a very famous effect in psychology, highly replicated in many different material, but that's the original curve that actually he himself studied, which basically shows a very simple effect, that whenever you remember something, you encode something, it can retain, it can be preserved in our, say, mind for a while, as you may see, right? Immediate recall, so most of the information is retained, but as a function of time, most of this information tends to disappear, right? This is it is called the forgetting curve. It has been studied, as I said, kind of heavily. It expresses, and it also shows very specific issues that I wanted to highlight here. The very first one is that not all the memories seem to disappear, because we can see there's some sort of like an asymptote here. Many memories, at some point, perhaps after a few hours, after a few days, those that can be retrieved at that point seem to be preserved over the course of time. So there's something specific happening at these memories, right? These seem to be something different for these ones that cannot be shown in the previous ones. People that have explored this idea that some of the memories may disappear and others may not disappear have actually found as well that this sort of like rate of forgetting seem to accelerate over the course of life. For instance, in these findings, they just simply showed, as you can see here, that comparing young and older adults, when they have to recover information that was studied just briefly a few minutes after encoding, the two groups of patients, two groups of people seem to recall severely well, right? And even after 30 minutes, but then there's a clear decrease of this preservation for all the adults, meaning that there's something specific happening at longer time intervals after encoding something that makes these people to forget in an accelerated mode. So if we focus on this sort of like delay period, one would like to see which are the protecting factors that preserve memories for a long time. And one of the few findings that was kind of like highlighted a few years ago already, as you see, right, on the 1924. This study was a very simple one, and they just showed these two individuals that memories tend to be highly retained just by the fact that in between the encoding and the recall, there's a period of sleep or there's a period of awake. This is a very simple phenomenon. That was the very first ones to report something like this. So basically these findings, what state, is that this sleep period that separates the encoding and recall seem to play a role. So over the course of this, over the course of the next years, people started focusing a lot on understanding the neural mechanisms that may play a role during sleep to protect memory forgettings. In here, I'm going to start by focusing on one of the most important ones that we know these days, and this is done on rodent studies. This is a very simple task for those who are on the rodent studies. It's a very simple one, behavioral one, where the rats or rodents, they need to navigate on a simple path. This is like an alternative path task, as you may see here, right? And what they can do, which is interesting for us, is that they can record single neural activity from the hippocampus. And what we know from a few years now is that then there's some specific neurons in the hippocampus that seem to code for the position in space, what they call the place cells. These place cells can be identified in the hippocampus, and the good thing is that you can see how these cells pick their activity at specific locations in the trajectory of the navigation space. As you may see here, there will be different cells picking, right, sequentially as the animal moves around the trajectory, sequentially. But what they found as well is that when these animals go to sleep right after this task has learned, is that the same cells reappear spontaneously and in the same order. Right? And this is a fundamental issue because it seems as if the brain during sleep seems to enter into a mechanistic mode where might be replaying experiences that you have just learned. So this ingredient, this mechanism was very relevant at that time because it basically slightly changed the perspective that we have from sleep. It's not just a passive aspect of our behavior. It seems to be very active and might play an important role. So what we wanted to know, and that's going to be our first study, what we wanted to know is whether something around these ideas may also be accounting for memory consolidation, say, right, avoiding the forgetting of memories. We wanted to do that in humans, but of course, as you may expect, this is tricky because in humans, we cannot just pick up single neurons or place cells, right, as you can do in animals. But we had to rise up with a strategy, I'm going to tell you now. So the very first question we wanted to basically ask was, is this hippocampus, this region that seems to be very specific in replaying just encoded experiences, relevant for consolidation through a mechanism that we have seen in animals that accounts for replay? So to do that, what we did is we selected a group of healthy participants, a group of patients that had specific lesions in one of the hippocampus, with the sclerosis in that case, right, as you can see it here, and another group of patients that had specific lesions in the two hippocampus bilaterally. But now the point is, how do we reactivate specific memories? How do we do that? In animals, you can track this back because you can record this activity, but in humans, you cannot do that, as I said. So we had to come up with a strategy, a bit indirect, but you see it's quite effective. So we asked patients to encode words with the specific sounds that appeared simultaneously. For instance, the word appeared on the screen, like truck, and they had to associate this with a specific sound. And we gave them several pairs of these elements. Once the patients and the healthy participants learned these associations, we asked them to go to sleep in a normal booth. Actually, it was in the hospital. Now, the key point is, how do we induce reactivation of specific memories? So the trick that we do was to apply something that is called target memory reactivation, which basically, it's very simple, essentially. It involves getting the sound, half of the sounds that were the patients learned during the encoding phase, and presenting them back again while sleeping in a specific phase, in a slow wave sleep phase. So the idea is that if this sound is heavily associated with a memory, we would induce some reactivation of the associated representation. And by replaying artificially this memory, we would expect these specific memories to be less forgotten compared to those that have not been replayed artificially. So the day after, basically, when they woke up, we just asked the patients to retrieve all the memories again. So these are the results for the healthy participants. So in green, you see the proportion of memories that were retrieved on day 2, say after waking up, right, that were reactivated through a sound, right? And in red, you see those memories that were not reactivated during the night using a sound. And we observed a minimal but substantial, or at least statistically solid, effect. So those memories that were artificially replayed by a sound were better remembered the day after. We found a similar effect, though slightly lower in general, for patients with one hippocampal that was lesion. But this effect completely disappeared on patients with the two hippocampal lesions. So we also found that the degree of the hippocampal spareness, accounting for the two hippocampals, correlated with the degree of benefit of the reactivation during sleep. Now, we wanted to jump into a slightly more mechanistic question. And what we knew from animals is that during sleep, and recordings on the hippocampus, there's a prominent activity that cannot be seen during awake time, which is this very slow and high amplitude waves that you can see it here. And what we know from these studies is that these very slow waves are very relevant because they seem to kind of like partialate the excitability state whereby the hippocampus may replay memories, and we know that. So in what we explored in patients is whether this slow wave activity as a physiological parameter was also different between patients and healthy participants. And we found actually that there was this gradient decrease of the amplitude, namely that, right, the more the hippocampus was impaired, the more the slow waves activity seemed to be also lesioned or impaired. So we show in this study for the first time that the hippocampus seemed to be very specific and very relevant to reactivate the memories on humans, and that this has an effect on the consolidation of memories. Now, if we want to move one step further, and we want to explore how this reactivation, how this memory consolidation phenomena is important to preserve memories in a daily life activity, we have to assume that, in fact, We are not just encoding things in the day life as if it was a list of words. For example, imagine that you just leave this place and you encounter someone like, right, this person that you may not know, right, with this dog, walking this dog, which can be represented something with this sort of like 2 notes. And the day after, right, you just come back to the clinic, hospital, and you see another person, like this one, walking the same dog. You may not know the 2 of them, but of course, I guess that you're all aware that we can make this sort of like rather automatic inference, assuming that somehow these 2 persons might know each other, right? They might be partners, they might be friends, they might whatever, right? But they seem to be closer than random people. So actually, what I wanted to emphasize here is that the way we learn things, the way we encode information, the way we preserve memories is associatively, right? We relate things. So the question is, if this is like the fact, which is something that we all experience, and we know that replay or reactivation during sleep seems to play a role for consolidation, we would like to know how reactivation of associated memories have an impact on consolidation. So to explore that, we just ran another study, which people had to encode images on a grid like this on a screen, and they were forced, or they were actually asked to encode the position of these images. On a subsequent stage, we asked them to encode one of the same images with a sound, and one of the images was simply like this placed to another position. So the idea is that these 2 memories share one ingredient, one element of the experience, but the other one is completely different. Because we also asked them to learn the association with the sound, right, as we did before. During sleep, we could manipulate the reactivation of these memories by producing again or introducing the sound. And then ask again on the day after how these memories can be, how these memories were preserved. So the very first thing we found was that we, by replaying the memories that were encoded on the day before, the memories that were associated were also preserved. So remember that in the previous study, we showed that by artificially replaying one memory, the same memory was reinforced or was boosted or was less forgotten. Now in this study, we show that not just the same memory is reinstated and is consolidated, but also the associated one, which is what we tested here. We show this effect is specific to the sleep. But here we have a problem. If we assume that any reactivation during sleep spontaneously has an impact on all the associated memories, we would end up having a very cluttered representation of our day life, right? We would have like memories that completely overlap to each other, and we would not be able to disentangle which memories come from what. So we need some sort of like degree of organization of these memories. We need to organize them somehow, and we need these memories, these mechanisms, to act accordingly. Otherwise, we would mess up all the representations. So that was a brief example. So we assume that one potential effect, or one potential factor that could prevent memory cluttering would be that during reactivation, somehow the neural network took into account the strength of the associated memories. And to test this idea, we just repeated the same experiment by asking the participants to learn the 2 overlapping memories by separating them a bit further in between them, assuming that when they encode a memory, the previous memory was less activated and therefore was more weakened. So, and then we asked people again to go to sleep and we reactivated these memories. So what we observe in this case is that whereas in the previous scenario, the reactivation of one memory has a positive impact to the associated one, now the reactivation of one memory had a decrease of the remembering possibilities of the previous one. So it was an opposite effect. The idea here is that somehow it seemed as if when you replay information, the replayed memory may also project to the associated memories, but the level of strength of these memories may determine whether you associate and you positively consolidate these memories or whether you weaken them, and that's something that might be relevant. So finally, I just wanted to bring one more example of how we can, the replay of the activities may have an impact on memories. And if we think about these sort of like experiments where we explored individual images or even like grid information. And if we think about how our experience unfolds over time, that's something that doesn't really fit, right? So the way we normally have an experience is unfolds sequentially, right? One element after the other. It has a continuous aspect. So if we want to understand how we remember things from yesterday, from a month ago, we need to also understand how our system is capable to segment this information that even though it appeared continuously over time in our experience, it remained fragmented or segmented as an episode at a longer time. So we had this hypothesis that the same mechanism that appeared during sleep, which is this neural replay, may also take place during awake encoding, during our experience. Here I just show like a very simple situation where imagine that you go to the kitchen and you prepare some food for your staff, and then right after preparing this food, you go and decide to go to the living room to watch your series, your favorite series. One could just easily glimpse this idea that maybe the day after or maybe 1 month after, one can maybe remember what they eat or how they prepared information by just forgetting as well what they watch in the movie, in the living room. So somehow, even though the experience was continuous, right, these two memories became dissociated. So our hypothesis is that somehow, during our wake time, when we shift from one episode to another one, our brain somehow rapidly wrap up this information. And this is not something that kind of like comes up like magically. In rodents, it's also shown that when the rodent is learning something, right, whatever, it's navigating in a path, and we can track these kind of like place cells movements, right at the very end, this would be the end, when the animal is resting, there's been shown as well that there's a rapid replay of the just encoded information, right? So it seems that this sort of like similar mechanism that shows up during sleep may also reappear during awake time, as if it was like a very fast consolidation phenomenon, something that allows us to keep this information kind of like glued together. So our hypothesis was, to make it short, we just wanted to explore whether at the very end of an experience that we could present on a computer, some of the elements, some of the neural patterns that appeared over the course of this encoding reappeared right at the very end. And for that, we had this sort of like analysis, which is called spatiotemporal pattern analysis, which basically, to simplify, it allows us to quantify the degree to which neural activity at a multivariate level resembles activity that appeared previously. The more the activity resembles the previous one, the more this activity might be related to a replay. And this is actually what we found in many, many experiments so far now. This would be like the time length of when the offset period starts, so when the episode finishes, and that would be like activity coming from the previous episode, and in red would be where this sort of like increase in reactivation shows up. These days, we're doing something similar with humans, via intracortical recordings, because we would like to link this with this ripple activity, these slow waves in the hippocampus. And just to finalize, because I think I'm running out of time, right? So I just wanted to conclude that the slow waves during sleep seem to be associated with hippocampal memory consolidation and replay. And even though that's been shown in healthy participants and in rodents, I think it's kind of like important to highlight that there's a strong line of research to link this with aging and perhaps Alzheimer's disease. This is like one of the examples of one group that showed that slow waves, right, this sort of like pattern that we explored in patients seem to decrease in amplitude, as if it was a fundamental mechanism that seems to be impaired as well during sleep, a general one. Now, the important thing about this slow wave as well is that there seems to be ways to enhance the amplitude of them again. There's a couple of papers now, very famous ones, but a few others, that shows that by simply like presenting some auditory clips, or even with TMS, by electrically inducing activity right at a specific moment of these slow waves, which seems to be specific windows of plasticity, right? It seems to have an impact on the memory as well. And that's been shown in all the patients, in all the population. And finally, just to finalize, this idea that actually it's also relevant how we replay, how we segment information during the course of our day life activity. There's some research that just came up showing that in all the adults, this segmentation process linked to the activity of the hippocampus, which is the one that we've been exploring in this talk, seemed to be slightly diminished, as if it was a segmentation problem in general for these patients. So I just wanted to thank, like my group here in Barcelona, some of the collaborators that participated in these studies I showed, and then also some of the hospitals I'm working with, or we're working with, relate to a patient study and the intracortical stuff. Thank you very much, and I'm happy to take questions.
Okay, thank you very much, Lluís. There are some time for questions, short questions. Nicolai.
Thank you very much. That was fascinating. I would like to come back to your initial study on that hippocampal sclerosis or temporal epilepsy study, where you showed that when only one hippocampus is affected, that you hardly observe this effect. But is there any modality specificity depending on the laterality of the lesion?
I think that's a very good point, though. In the task, we use visual images together with sounds. So at the end of the day, it was a bit of a mixture. The unilateral were a mixture of patients. Most of them were, I think it was back a few years ago, I think most of them were left dominated, I think. I'm not sure. On the other hand, this sort of like dominance, right? I think it's also a debate, no, on its own.
Yeah, absolutely.
Agree. That's a good point.
It's
I think we explored this in the data. It didn't really show up as something that changed the effects, but I'm not fully remember that.
And do you think then that maybe one hippocampus depends on how long that patient has had that lesion?
No, that's a very good point. That's
Takes
absolutely.
over the function.
Absolutely, absolutely. So for these patients, we took like chronic epileptic patients where, right, I mean, they've been with this disease for a few years, even longer than maybe the clinicians may know. So certainly that's an issue, right? The plasticity phenomena that basically took up during the course of the years for this may have an impact as well. It may also explain why unilateral may kind of have this function preserved. That would be another possibility. Yes, that's a good point, yes.
Thank you.
Yep.
Another question? There.
Thank you very much for your inspiring talk. I have a question regarding your experiments on recall and association between those words and the sounds in the patients. Have you seen any correlation on that recall of those memories with, for example, the quality of sleep or the number of hours of patients?
Sure. Yes, that's another very interesting aspect of the data. We observed that slow wave sleep, spindle, if you are into the field, spindles, which are another neurophysiological response that is heavily linked to consolidation, these factors seem to, actually correlated with the memory improvement related to replay. In many other experiments now in humans, this is because it's something that's kind of like easily collected on scalp EEG, for instance. There's been that's kind of like long evidence, at least now I would say, that the quality of sleep, depending on the functions, do have an impact on the memory quality, though it always depends on the measure you're taking, right, and other aspects. But it seems to be a strong link between the two, yes.
Thank you.
There are another question, I think.
Hi, thank you for your talk. I was going to ask you if you have done any study also in clinical population, like the post-traumatic stress disorder.
No, not ourselves, unfortunately. I mean, the sleep studies are kind of like complex, right? You really need kind of like big infrastructure. I'm just trying to think on other possible studies, but I'm not aware of any. I know people are approaching this from the emotional point of view, and you know, the problem with this is that at the end we induce reactivation, right? So inducing reactivation means that somehow you may want them to re-experience stuff, whatever, even if it's like kind of like, let's say, a representational level. So for like clinical patients, then this might be ethically problematic, right? Because, yes, right. That's one of the reasons some of the like stress disorders, things like that, are kind of like set aside a bit of these sort of like studies to some extent. There's a lot of research though, and a lot of interest, and a lot of like investment in developing, let's say, artificial strategies to induce forgetting through reactivation of competing memories, for instance. Another possibility would be to artificially induce specific time windows where forgetting may take place. And this is done electrically stimulating like the amygdala, for instance, and on the hippocampus. But that's a slightly different story. It's a bit indirect, right?
Okay, thank you.
Okay. Yes. There are no more questions. I have a question, but I think that you answered now. How we can to make the intervention in these slow waves to consolidate the memory? What is the strategy that you are thinking about?
Yes. So at least the ones. So I just wanted to kind of like emphasize these results because I think are kind of potentially very relevant, which is. So the slow wave. I didn't enter into the mechanism itself, but the slow wave activity, the slow waves, are very slow in essence. So it gives us some time to intervene on the one hand, and it's shown in animals, in models, that is a plasticity window. It's kind of like very prominent in the cortex and subcortical levels as well. So just to say, it seems to be a very optimal time window to start with doing stuff. And so these results that actually they've been shown like a few years ago, what they just showed is that by simply presenting an auditory click, like click, on the right moment of the sleep wave, which is in that case in humans was the positive, the up state, say, just this had an impact, a beneficial impact in the memory retention. So it's kind of like prominent. Yeah.
Very interesting. Yeah. Thank you very much, Lluís, for this interesting talk. And we move to the next one. Okay. The next speaker is Raquel Sánchez-Valle. She's head of the neurology service in the hospital clinic and assistant professor. She belongs to the DBAPS and also to UBneuro. Her research group is Alzheimer's disease and other cognitive disorders and focuses on the use of different types of biomarkers from neuroimaging to genetic biomarkers for the early and accurate diagnosis and prognosis of neurodegenerative dementia. She will present the talk Catching the Glimpse of Alzheimer's Disease Brain.
So good afternoon, everyone. Thank you for the invitation to present. As you can guess, based on my white coat, I'm a clinician. So I have the doubt if I should present you some set of the papers or the studies we are working in our group. But afterwards, I thought that maybe that will be better. I will briefly summarize in which page the clinicians are in this in Alzheimer's disease, because sometimes I think that clinical and basic scientific, sometimes we are lost in translation when we talk of the same thing, looking from different perspectives. So I will thank the previous speakers because they have summarized some of this evidence here. So, as you probably already know, in 1906, Dr. Alois Alzheimer described a clinical case of this person over there that was Mrs. Auguste, who developed cognitive and behavioral problems at the age of 51. The neuropathological examination of this patient showed brain atrophy, neuronal loss, and the presence of extracellular senile plaques and intraneuronal neurofibrillary tangles. And this is the original drawing from Dr. Alzheimer's. This clinical pathological entity was several years later named Alzheimer's disease after Dr. Alois Alzheimer's. It took some time, but in 1984, the first clinical criteria for Alzheimer's disease was published, and this clinical criteria say that only the neuropathological diagnosis could confirm the diagnosis of Alzheimer's disease. And they say here, you can say that this diagnosis cannot be determined by laboratory tests at that moment. And they say that this criteria will be reviewed as more definitive information becomes available, but it took about 30 years for this criteria to be reviewed. So that we will discuss why. How good were these criteria? These criteria for probably AD definitions achieve either good sensitivity for AD at the expense of specificity or vice versa. So it was around 80 versus 70 or even with a broad range from 50 to 100. So quite different between the different cohorts, but not that good as we expected. Afterwards, we know that the 20 to 25 patients included in the first anti-amyloid drug trials did not have amyloid plaques in their brain, so they are receiving an anti-amyloid drug when they didn't have amyloid in their brain. So it was clear that new criteria and biomarkers were needed. So we knew that the atrophy in Alzheimer's disease was clear, and the typical area of Alzheimer's disease was the medial temporal lobe. So we first us as neurologists start looking at the MRI at the temporal lobe, medial temporal lobe atrophy. But it was quite good when patients came to our offices at this stage, at moderate stages of the disease, but was not so good when patients start coming to our offices in this stage, in early or somewhere between A and B. So it was not so clear at the visual examination if the patient had or not had a medial temporal atrophy. And this, for example, in this paper from one of our postdocs, Neuropharmacology, showed that the MTA atrophy at the level of 1 showed just a sensitivity of 63% and a specificity of 81%, comparing early onset Alzheimer's disease versus control, but were not relevant, was not significant difference between Alzheimer's and another type of neurodegenerative dementia that's called frontotemporal dementia. So probably not well enough to be used in early stages of Alzheimer's disease for diagnosis. So just after these criteria were published, we learned that the extracellular plaques were composed by beta amyloid. It was after that criteria, not before the criteria, and that these intracellular neurofibrillary tangles were composed by hyperphosphorylated tau. These are images from our neurological tissue bank that's in the basement of this building, and where we have about 2,000 brains from different types of neurodegenerative dementia that are available for the scientific community. So after we learned that this A-beta and tau were the main composers of these neuropathological hallmarks, we start looking for tau species and beta species in CSF in the 19th. In the beginning of the 21st century, we have enough experience with these markers, with this huge amount of samples in a meta-analysis, including some of our center, and we knew that the total tau, the NFL, neurofilaments, the phosphotau, and the E-beta were good markers to differentiate Alzheimer's disease from controls. And we create the CSF-AD signature, that's low A-beta and high total tau and phosphotau levels in CSF, with a sensitivity and specificity about 85 to 90% to differentiate Alzheimer's disease patients from FTD and controls. And this signature has a strong association with neuropathological change, amyloid plaques, and NFL and neurofibrillary tangles in brain-confirmed cases. Afterwards, we knew that discovered traces that could discover or mark amyloid in the brain in living patients. This is anti-amyloid. That is the typical diffuse pattern of cortical pattern of amyloid tracer uptake in Alzheimer's disease patients. But for example, in this study with the group of Salayong and our group, we also can see another type of amyloid patterns, especially in genetic cases. But with these markers, we can track the amyloid deposition in living patients, and these are commercially available and approved both in the FDA and by the EMA, and available in Spain, in our center. We have also the tau marker that have been discussed before. This is approved by the FDA, but not by the MDAA, so we cannot use this tracer in the clinical setting in our country. But as discussed, the tau PET trace uptake marks the presence of neurofibrillary tangles in brain, but the tau traces uptake shows a strong regional association with clinical and anatomical heterogeneity in Alzheimer's disease. So we can have this typical pattern if Alzheimer's disease, but these different regional patterns depending on the clinical presentation of the patient, even if all of them are Alzheimer's disease patients. Sorry. So 30 years later, almost 30 years later, the National Institute of Aging and Alzheimer's Association published this research and clinical criteria in 2011 that include biomarkers, amyloid beta markers, CSF or PET, and also marker of degeneration. In this study, they include CSF tau, but also FDG PET and MRI as markers of Alzheimer's disease. But 10 years later, the International Working Group published another set of criteria that exclude MRI and FDG PET and only include amyloid markers and tau markers, both CSF and PET. And these are the current research and clinical criteria we are using. One thing is these criteria include not only dementia, but also MCI stages of Alzheimer's disease. For us, MCI patients are not healthy aging, they are considered patients, and include both amnestic and non-amnestic presentation of the disease that were not included in the previous set of criteria. And what is the impact of this change, including biomarkers in our clinical practice? That is quite high, I think. 25% diagnostic change, 14% of increase in diagnostic confidence, and 31% change in clinic management of our patients. So we think that biomarkers are needed and have an impact in our clinical practice. And what are we now? So CSF is quite invasive, even if the lumbar puncture are well tolerated. It's not as well tolerated as a blood-based biomarker, and PET scans are expensive, so it cannot be used in every patient. But now we are looking for blood-based biomarkers in Alzheimer's disease. The new technologies provide the opportunity to identify and quantify 80 biomarkers that are present in blood at a very low concentrations. Several companies are developing new blood-based biomarkers for Alzheimer's disease, for example, p-tau, NFL, plasma A-beta, or GFIP, a gastroglial marker. And this is our study, for example, in our cohort, just recently published, that you can see here that Alzheimer's disease patients present increased levels of p-tau, and when compared to cognitive unimpaired or non-neurodegenerative disorders, we show an area under the curve with 0.990 something. So this is quite accuracy for diagnosis in living individuals. Other type of markers like NFL are good markers of neurodegeneration, but are not specific enough to differentiate Alzheimer's disease patients from other type of neurodegenerative dementia. So as previously shown, these biomarkers also provide the opportunity to study Alzheimer's disease before the clinical onset for research purposes. Previously, the previous speaker showed an image from Jack and collaborators, that's from sporadic Alzheimer's disease. And here's the stages, the same type of study done in dominant inherited Alzheimer's disease. That's the genetic form of Alzheimer's disease. With all these markers, you can predict how far from the disease onset is a patient that have inherited the pathological mutation. Okay, and also the AD biomarkers can be used as endpoints for disease-modifying drug trials. For example, this is the results with aducanumab. We can discuss if aducanumab has impact in has a meaningful clinical effect. You can discuss if 0.5 CDR is meaningful or not. You may also think that, for example, if the drug has a magnitude effect of 100%, or complete effect, the placebo group just evolved in 1.5 CDR sum of boxes. So it's the difference of 0.5 is about 1/3 to 25 to 1/3 the magnitude of effect compared to placebo. And we can discuss if that's a lot of effect or small effect, but we cannot discuss is that aducanumab was the first drug, the first anti-amyloid drug that show strong evidence that he can clean the amyloid for brain and that have an effect not only in amyloid but in other markers like p-tau, NFL, and other downstream biomarkers in Alzheimer's. And we have also participated in the study, and we have also participated in the study with dominant Alzheimer's disease network, that's with gantenerumab and solanezumab. Solanezumab did not show any effect, neither in clinics and neither in biomarkers, but gantenerumab showed an effect on biomarkers, both in amyloid markers and downstream markers like NFL or tau markers. When we say that the gantenerumab has not any clinical effect, we must think that the symptomatic patients, genetic symptomatic patients, are very advanced. They have no changes compared to placebo, but the asymptomatic, non-symptomatic participants that were the main focus of the study didn't decline, neither in the placebo, neither in the active arm of the trial. So we don't really know if this drug changes the natural history of these participants. Now we are in an open level stage of the study, so these participants are receiving gantenerumab, and now we are moving to another trial that will combine both anti-amyloid drugs and anti-tau drugs. We can think or discuss if anti-amyloid drugs have, as I said before, a minimal clinical effect or not. But from clinicians, it's difficult to think that any other type of drug will work having this enormous amount of amyloid there. So we think that even if the effect of the anti-amyloid drugs are not strong based on clinical performance, may be needed to be combined with other type of drugs to have a meaningful effect, because all these correlations between amyloid, tau, and inflammatory markers and other markers. So we probably think as clinicians that anti-amyloid drugs are not enough for our patients, but probably we will need to have anti-amyloid drugs to modify the natural history of the disease in our patients. So take-home message, AD biomarkers provide the opportunity to identify neuropathological changes, especially amyloid plaques and neurofibrillar tangles in vivo. Current Alzheimer's disease clinical and research criteria include biomarkers, biochemical and neuroimaging markers. Clinical diagnosis based on clinical criteria only is still valid for a relevant percentage of patients, but has limited utility in a research context. Blood-based biomarkers are under investigation, might become useful as a screening and/or diagnosis tool in few years. Neither of these blood-based biomarkers are approved, but could be probably approved in few years. AD biomarkers provide the opportunity to investigate brain changes in Alzheimer's disease before clinical onset, and also provide the opportunity to investigate the effect of disease-modifying therapies. So thank you, this is my group. Thank you very much.
Okay, now we have time for two short questions. Okay. If you have to choose an actual known biomarker, or what do you think that is the best to follow the drug, for example, the drug effectivity or the progression of the disease?
I mean, in fact, these drugs monitor the brain changes, but not really are proved to be markers of disease progression in the sense of prognostic. So the most probably could be NFL, but it's doubtful their effect as progression markers. But of course, I think that depend on the drug. I mean, you must. You need a marker of target engagement. So if you are testing anti-amyloid drugs, you will need some PET marker or a CSF marker of amyloid, and another markers to see what happens in downstream markers below the amyloid cascade. Of course, if you are targeting tau, I mean anti-tau drugs, you will need an anti-tau, a tau marker. For example, in clinical trials, we are using usually tau PET for anti-tau markers, anti-tau drugs.
There is not a unique answer. Okay.
No, they are different depending on the type of.
Thank you very much.
Drug. Okay, thank you.
Next. I now give the floor to Jesús Rodrigo. It's our pleasure to receive Jesús Rodrigo. He's Chief Executive Officer of the Confederación Española de Alzheimer, but in addition, he's board member of Alzheimer Disease International and also President of Alzheimer Iberoamerica. Of note, he's member of the National Dementia Group in charge of defining and drawing the National Alzheimer Plan 2019-2023. And he's actively participating in numerous projects and consultations, both nationally and internationally, and for sure he gives us a new vision of brain and Alzheimer's disease in particular.
Thank you very much for the introduction. The pleasure is mine to be here to share with all of you another perspective, another vision, not about the brain, but the effects of the disease of the brain in person, in people. This may be one of the most important speeches I can take, because talking to people who is in charge of research, diagnose, treatment, and so on, is very important for us, as an organization of family of people affected by Alzheimer's disease. Of course, I can't teach you anything, but I can share with you some reflections, some things from experience working with people affected by Alzheimer's disease and their family. This is obvious. The brain ages just as the person does. As the rest of the human organs. It's a pity that we are celebrating new birthdays, so not only the person, but all of our organs do. But aging is not synonymous of disease. There are a lot of people with a lot of years, very, very healthy people. Hopefully, the disease is only a part of human beings. So it's not a problem to get older, but you can have the luck or not to have a healthy life or a diseased life. But having the disease is not the same to be sick. So I can have the disease, but be a perfect and normal person. All we know that one of the most important and prevalent disease of the brain is the dementia, is is Alzheimer's disease. About 91% of dementia cases is in older people over 65 and more. And of course, these people is in which we give all our attention. So when you talk about a person with dementia, the first image which come to us is old man, old woman sitting in a chair without any kind of movement, without any kind of reaction, and it's almost a vegetal person. Okay, this is the first image they come. But and of course, research, all attention, all support from the health and social systems goes to these people. The main part of this attention goes to this. But this is the 91% of the cases. But some years ago, the World Health Organization said that about 9% of all diagnosis was are younger than 65. Younger than 65, and thanks to the advances of research and diagnosis, people diagnosed in the very first stages of disease. So this, I think we can clap this finding because it demonstrates the importance of research. Here, I would like to present you in this picture part of our panel expert of people with Alzheimer's disease. Can you recognize who has the disease and who not? No, perhaps not. Perhaps not. It's difficult to know who is sick and who is not. Well, these people, these new people, these young people affected by Alzheimer's disease in the very early steps of the disease has new needs, has new interests, and we are not ready to know that. We are not ready to know that because our attention goes to the other 91%. But these people going to or associations looking for an answer to their needs, they can't find because we are not ready to give them or to offer the answers they need. So the first thing we must have in mind is there are healthy and aging brains. There are diseased aging brains, healthy young brains, and diseased young brains. So this is obvious again. This is obvious again. But I would like to share with all of you 3 ideas which are key for me, which are key for my organization, the Spanish Confederation of Alzheimer's Disease. First of all, prodromal or early research and diagnose. Diagnose is the key point attending people with Alzheimer's disease. We'll see after that. The second idea is we need to understand the needs and interests of this new group. This new group, some years ago, was 9% only, but thanks to you, thanks to the development of research, in the next years, this 9% will become 12, 15, 20, I don't know. I don't know. But I think this is a very important and very interesting aim for all of us. And the third key idea, which is really important, is we must fight against social exclusion. There are a lot of times that the early diagnosis create exclusion of people with Alzheimer's disease and their family. So we'll see it after, but we need to keep in mind the importance of non-exclusive societies, not inclusive societies, not exclusive societies, because you can't include in your community people who has been an active part of this community during all their life. Okay. Why is important for us, from our point of view, the research? Why is important from our point of view the need of an early diagnosis? Because despite the reluctance of many professionals, so why we are going to give the early diagnosis, if we have not the necessary tools to offer from the pharmacological point of view? Okay, that's right. There is not any tool to offer people with Alzheimer's disease. We have not any drug to combat Alzheimer's disease or other kind of dementia. But if you provide the family with early diagnosis, the family gains time. And this is very important for the family. Time. Quality time. Time to affront the disease and the process they are going to front. Time to take decisions. Time for the person diagnosed to decide how he will want to be cared. How he will want to be cared. All necessary decisions must be taken in this period. And it's important to ampliate this quality time from the family point of view. Independent that all we know that the disease is a trouble with not a stop. So keep in mind, please, prodromal or early diagnosis. Don't think you have no tools to offer. Think in the family, they gain time. The second idea, we need to understand the needs of the new people. This new collective, this 9%, this is a publication of the last year made by our PEPA, expert panel of people with Alzheimer's disease. These people sit around the table to share between them their expectations, their expectatives, their needs, how they feel with the disease, how they feel with their family, how they feel with the society, how they feel with the health system. This is available on the website if you want to read it. But one of the very interesting thing they said is, what's the difference in me when I wake up in the morning to go to the doctor looking for the diagnosis? They said me, I have Alzheimer's disease, and go out of health center. Has been any important changes in me, or I'm still the same person? This is one interesting thing you can find in this publication. So, how can we treat a group of people if we don't know what they need? Remember, all of efforts are focused in the 91%. This 9% is growing. So the profile of people with Alzheimer's disease in the short term is going to be changed. So it's important to not to ask. It's important to learn from them. In the rest of diseases, it's easy because people maintain their conscience and they can take their decisions. In dementia, it's the first time that people with Alzheimer's disease, people with dementia, can express their needs, their desires, and so on. And that is thanks to the research, of course. And the third idea, and this is my last point, is fight against exclusion. Fight against exclusion. Our societies or communities must be friendly with people with Alzheimer's disease and other kind of dementias, not only with the person who has the disease, but with the whole family. This is a binomius, people affected by Alzheimer's disease and family caring that. So it's very important that society be conscious of this new profile. In this picture, you can see the members of the expert group of people with Alzheimer's disease in the same table with the mayor of Pamplona and the Regional Ministry of Social Affairs of Navarra, presenting the document I showed you before. So these people has the disease. Are they sick? This is the question, and this is, I think, a good opportunity to think and to take new considerations and take new decisions. So many thanks for your attention. Many thanks to the Institute for inviting us to share with all of you those messages. I think this kind of forum are, from our point of view, very important, and I need you to know that the hope of almost 5 million people in Spain living with Alzheimer's disease, between people with the disease and their family carriers, those hopes are deposited on you. Thank you so much.
Okay, thank you, Jesús. Thank you for offering us your vision, the vision from the other side of the mirror. And now we have time for questions or commentaries.
Okay, thank you. This is very, very nice and very interesting. I have a question about, because from the research point of view and scientific point of view, is to have this early diagnosis is very interesting. But from the patient, it's good to have this early diagnosis if you cannot do anything. I mean, if you don't have symptoms, because we are working with Huntington disease, and this is a clear genetic test that if you get the mutation, you have it sometimes, no? And many people don't do the test because you are afraid to have it. And from the patient point of view, do you want to have this early diagnose, or is better to, I don't want, if I have symptoms, I will do it. But
this is a question afraid all of us. But when we create the first expert panel of people with dementia in 2017, one of the first questions I made them was this: Do you appreciate receive an early diagnosis? And the unanimous answer was yes. Yes, because I have time to enjoy with my family. I have time to affront my disease and to be conscient of this disease. I have time to put all my matters in order. I have time, quality time. But not only time for taking decisions and so on, but I have time to, to participate in different kind of supports from the point of view of non-pharmacological therapies. So this makes us, they said, make us still value for my family, for society, for my group of friends, for my neighborhood, so on. Give me strength to do things. Give me strength to go, to keep away. So it's important for these people, yes, of course. But not only is important, but one of their concerns is to show the rest of society they still are a valid people. So they are not all, okay, but an important number of people from the expert panel is so ugly because when they receive the diagnosis, automatically they receive the hiding from the job. I want to keep on working. I can working. Of course, not in the same position, but please adapt the competences, adapt the responsibilities. I still can contribute to the society and to the company. So, answering your question is yes.
Very much, Jesús, for showing the position of this population, especially for the early PEPA, for the early Alzheimer's disease population. And now, especially in the clinical community, we are discussing about the fate of drugs, of these anti-amyloid drugs. And it's clear the position of the Alzheimer's Association, the American Alzheimer's Association, about aducanumab and lecanemab. But what are your position, I mean, from the Spanish point of view? It's 25% of more time in the early stage enough to for you to take a drug. We will not discuss the cost, but just to support this drug, or will not 25% of time in the early phase, more time in the early phase is enough effect?
Of course. If you have your son with a very dangerous and problematic disease, and a doctor says it has only 1% of possibilities, what do you do? What do you do? Of course, for us, it's really important those advances. In fact, when aducanumab came into our memories, we clap. Was the first drug after more than 20 years in the market. And we could clap consciently because one of the members of our expert panel took place in the clinical trials of aducanumab in the south of Spain. And when this person came into the trial, was apparently in an advanced phase of the disease. After finishing the trial, can read, can write, can drive, can do a lot of things she stopped before. So, 25%, of course, is important. For us, it's a very important advance of science. Of course, we know that new drugs will not be effective for all the people. This could be a miracle, of course. But I think more than 20 years after the last drug, if a new comes into the market and can't delay 25, 27% of the time of people with dementia, I think it's not only important, but can be inflection point to keep on researching. So, of course, if in the next days, the FDA or the EMA approves the new drugs, we'll clap with all prudency, of course, because we know this is not the definitive answer, but it's one more important step getting the answer.
Thank you.
Okay, we left here your talk. Thank you very much, Jesús. And you can clap also.